guys imagine, yes, you are into the spacecraft. Imagine you are going to the space. This is how the space will look like. Okay, since you guys know that the area is microgravity, so when you come out you can just float. But when you are floating, definitely you will be exposed to so many types of radiations. It may be gamma radiation, UV radiation, alpha radiation, beta radiation, whatever the radiation is there in the world, you will be exposed, isn't it? Now, do you think there will be any change in the gene expression of your body? Okay. So if you think there will be some change in the expressions of the gene in the body, then the next question is, how will you analyze it? Come on guys, how will you analyze it? Any methods you guys know? Protein analysis by Aarti. Protein analysis is not a technique, Aarti. Visible characteristics. What is that, Nitish? How will you do that? Why you want the expression vectors here, Sainika? Gene sequencing. How, Litika, you will remove each and every protein or gene from the person who went to the space, kill him, take each and every cell and then do sequencing? RNA sequencing. After that, what you will do? RNA sequencing. First of all, RNA is unstable. How will you sequence it? Tissue sampling, then analyzing. My question is, how will you analyze, Mamita? I know that you want to analyze protein. My question is, how will you analyze those expressed protein inside the body? Aarti says microarray. Vanshika says through microarray, 3 question marks. Can we go for blotting techniques? What is that blotting techniques concern? Since guys you will be after clearing CSIR exam, okay, even if you clear with the rank number 1, rank number 2, definitely you guys have to attend some interview. Okay, the best answer for any question which you don't know is I don't know, instead of saying something else. If you guys told me no, we don't have an idea, I must have gone to the next slide. Mamita Paul, very good. Compare amino acid sequence with new one. Definitely the gene expression is going to be different. Amino acids are going to be the same. Again, my question is, how we are going to do that? Okay, guys. First thing, if I want to do any gene expression, first of all, I want to measure the RNA levels of all the genes in the genome, isn't it? Because if any gene has to express, it has to convert from DNA to RNA, RNA to protein. Okay, so steady state RNA level can be measured by cell type, developmental program, external stimuli, and then moreover the time and the location plays a major role. That is a steady state we must have. But the genomic expression, the exons, it can be done by microarrays. Two people told, one person is with question mark. So microarrays is a method which we can go for global gene expression. This microarray is based on hybridization method. Another method to analyze gene expression is real-time PCR. So here I want to explain you measure what is meant by hybridization. Hybridization, it is binding of genes, that is a binding and RNA. This binding I am calling it as hybridization. It is same like what you guys can see in the southern blot or northern blot. So mostly this binding will be complementary between two strands. It is similar principle with RNA blot. So northern blotting, anybody has performed northern blotting, guys, here? No. So northern blot is a method which is used to study the expression pattern of specific type of RNA. Very important, you cannot study more than one or two, only a specific type of RNA. Okay, first of all the RNA has to be isolated and then it has to be transferred to the membrane. Then we want to probe with the labeled DNA complementary to the sequence, gene of sequence. Okay, so here the problem is the result may be visualized through a variety of method based upon the label used. It can be your fluorescent label or it can be your radioactive label. Okay, but here it is used to study when and then how much gene expression is occurred, and then moreover only one or two samples can be done. So if you see here the same thing, so this is what your RNA has been isolated, the first step. This is the first step, the RNA is isolated. After that the RNA is separated. After separation it is transferred to a nitrocellulose membrane. After transfer we will be probing it with the any complementary sequence which you are interested in. It can be with a radio label or fluorescent label, and then you'll probe it. The complementary sequence can be seen through auto radiogram. So maximum here I can see only one gene is expressed or not. Guys, can you understand what I'm saying? Are you understanding what I'm saying here, guys? Can I have response from the entire class, including those who are sleeping? So here basically what I'm trying to say here is, if I want to express, if I want to analyze any gene expression, the best method is, the available method is northern blot. But the problem here is I can analyze only one gene expression at a time. That's it. Okay, so these are the issues. So what are the problems will happen during this northern blot means? As I told you earlier, the stability issue with RNA. It is very, very unstable. During isolation of the RNA, if the isolated RNA is degraded, then your hybridization will not be quantitative. For example, how I can say it now? Let us take, you are growing a bacteria under 60 degrees Celsius and then 37 degrees Celsius. So now you have isolated your protein, your RNA. Okay. So let us think that some extra protein is expressed when you grow bacteria at 60 degrees Celsius. Let us think this is the RNA which is extra synthesized when compared to, while isolating, if this RNA is degraded, you will conclude that whether you grow in bacteria in 60 degrees or 37 degrees, both are going to produce the same type of protein. Will I conclude or not? Which is totally wrong. Understood or not? If this particular RNA is degraded. So that is the issue number one. And then number two, during hybridization, the probe should be properly standardized. If very less probe is there, then also signal will not be there. If it is too much, then also over exposed. This is the point number two. Hybridization here is we want to give enough time. If you give less time for hybridization and then wash it, then it will not go and bind, so you will not find any signal. So that is also issue, that is issue number 3. Number 3 is loading control. If some sample is overloaded, then also it will show overexpressed. Understood? Guys, are you following me what I'm saying? Okay. So in order to overcome the issues which are faced by the northern blot, where we can analyze only one gene, microarray came into picture. This microarray is global gene expression. At the same time, one shot. See, for example, in the RNA, northern blot, I can analyze only one type of gene. But in the microarray, I can analyze more than 1 lakh gene at the same time. Okay, that is the idea. So northern blot, only one gene at a time. It has some issues which I will explain. So advanced method over northern blot is microarray. So we are going to see what is microarray, how it can be performed, what are the advantages, how to analyze data using microarrays. Okay, you guys are ready? So what is DNA microarray? Question number 1. So this microarray, you guys know that it is also called as DNA chip. It is a method which allows the measurement of gene expression from a genome, in a genome, same time. Transcription, we know that it is a process of copying DNA into messenger DNA. Totally it is an environmental development. So a microarray detects mRNA rather than stable cDNA. Okay, so let us have an idea about what is microarray here. So initially this microarray require a substrate. Yes, Arthi. So the gene which we need to analyze are to be immobilized on a substrate. So sometimes it can be done using, initially it was done in the nylon membrane and then it was replaced by glass slide. Nowadays it is replaced with gene chip. It is 1 centimeter by 1 centimeter. Okay, we are going to explain that one. So if I just expand this gene chip array, you can see that there are so many immobilized immobilized DNA. If I just one spot, if I expand, you can see that the DNA which is immobilized. This is how it is going to be. So basically how it is going to be now. So in this slide, so there will be one circle. It will be like all of them will be done by robot. So here the DNA will be immobilized. Here one DNA will be immobilized. Here one DNA will be immobilized. This will be gene A, gene B, gene C. Everything we know. Understood or not, guys? You are following what I am saying? So each and every spot we are calling it as probe. So more than 200,000 different probes can be immobilized just within 1 centimeter by 1 centimeter probe. Okay. So now the basics of microarray. So now you guys know that if I want to perform a microarray, first the DNA has to be attached to the solid support. The solid support can be a glass, plastic or nylon. Okay. Next, here the RNA has to be labeled. Okay, usually indirect. So basically here we will use cDNA. I'll come to that point later. So now actually this labeled RNA will go and bind to the complementary DNA. Okay, that is what going to happen. That is what we will be seeing here now how it is. Guys, can I have a response from everyone? Everybody is following what I'm saying. I got response only from Mamita, Arti, Gayatri. That's it. Once more from where, Gayatri? Okay, nothing is that worry. This only I am giving outline. I will explain each and every step in the next slide. Okay, just wait. Can you wait for another 5-10 minutes, Gayatri, and then Avinash? Or else shall I go to the last slide now itself? So let me first talk about sample overall outline I'll give you. This is your normal cell. This is your diseased cell. Okay. So from both I am going to isolate the mRNA. Okay. So this mRNA is converted into cDNA. Gayatri, this is the steps. While converting, this cDNA will be labeled with fluorescent dyes. So usually the control sample will be labeled with green color dye, and then the sample will be labeled with red color dye. And then both the cDNA will be flooded on the microarray plate, and then it will be read using confocal microscope. This is the outline of microarray. So the sample can be tumor versus normal tissue, drug treatment versus no treatment, embryo versus adult. So now I can see what is that, what are the genes expressed in the normal tissue versus tumor tissue. What are the genes that are expressed in the drug treated cells and then cells without treatment. And then what are the genes which are expressed during embryo, what are the genes that are expressed during adult. Okay, understood or not? You are following what I am saying or not? Each and every step I am going to explain in detail. Yes, on the same microchip. Both the samples we are doing everything on the same microchip. Again the same thing I am explaining here. Cells from condition A, cells from condition B. I am isolating the mRNA. The mRNA is converted into cDNA. While converting, I will be labeled and then mix it and then overlay it on the microarray plate. Clear? So now already we have seen how to isolate mRNA in the previous classes, isn't it? You guys remember that? Isolation of mRNA from the cell. The same method is used to isolate mRNA from the cell. So once you isolate mRNA, this is your mRNA. So using an enzyme called reverse transcriptase, I can convert mRNA into cDNA. Okay. Exactly, Mamita. Okay. So while converting RNA to cDNA, during conversion, what we are going to do? So if you see that RNA, if I use a reverse transcriptase, it is going to produce my DNA, right? During that, so in the RNA part, wherever G is there, what will be added? C will be added, right? So those cytosine will be labeled. For your controlled cell, you will be using a green dye. For a sample, we will be using a red dye. The cytosine alone will be labeled like this. You guys have any idea why C is labeled? Why not T or A or G? Any idea, guys? Mostly the exons, that is the coding part, will have more GC content. Okay, when compared to G, labeling C is easy and then more comfortable also, more stable also. Okay. So as I told you, the green dye name is Psi3, the red color dye is Psi5. So it will go for competitive hybridization. What is competitive hybridization now? Just imagine here. So as I told you, green is control, right? red is deceased. Okay. So just I'm taking one, or let me take only 3 DNA. This is gene A, gene B, and then gene C. This, if the gene A is present only in the controlled sample, then the mRNA from control will go and bind. It will fluoresce green color. Okay. If the gene B is present only in the disease, it is going to be the red colored mRNA is going to bind. If gene C is present in both, maybe mostly it will be a housekeeping genes which will be present any time. So both red color also will bind, and then green color also bind. So this type of binding is called competitive binding. And then one more thing, the more complementary will have a more type of binding. Yes, Gayatri. So this is based on fluorescent. The next one, sometimes people use biotin also in order to label. Okay. So binds fluorescently labeled avidin. So mostly that is used for your Affymetrix gene chip, which we are not going to discuss here. You are going to discuss only the basic one. Okay. So now let us see the spotted microarray hybridization. What I explained. Control is labeled with green, and then red color is your deceased. So both the samples are hybridized on the microarray chip together. So wherever the complementary is there, it's going to bind red color red, green color green. If it is present both, and then it's going to be RG. So it's going to be green R, green R, green R, RG, 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 R, green, green, green. So what is mean by R here, guys? Pruta, what do you mean by this? Very good, Likita. So red color red means this DNA is present only in the deceased condition. Very good. Green, which is present only in the controlled condition. So RG means it contains, if the gene is expressed both the disease condition as well as in the normal condition. Okay, understood? So after probing, the slide will be kept under confocal laser scanning microscope. So now the laser will hit each and every spot, and then it will fluoresce the dye. The fluorescent will be traveled back, and then it will be detected. Understood? Guys, can you understand this? Can I have a response from everyone? So here the labeled dyes are Psi3 and then Psi5, where Psi3 is green, Psi5 is red. So after reading, this is what you will get. Okay. Here you are getting 3 types of fluorescence. One is green color, red color and then yellow color. Here you see lot of yellow, which means a mixture of green and then red. If it fluoresces together, it will give you yellow. So all the yellow color spot means this is the gene which are expressed under both condition. So nobody is cared about it. But red color, which means that it is expressed only during diseased condition. Green color, which is expressed only in the normal condition, controlled. So these genes are called biomarkers. So if you go to any doctor, if a patient is having disease, let us take HIV. So they will perform Western blot or ELISA against a specific protein GP120. Got it? So the GP120 protein will express only in the diseased condition. This is how they will find the biomarkers. Yes or no? Understood or not? Can I have response from everyone? Okay, so this one, for example, the RNA from the irradiated cells. Coming back to our astronaut story. RNA from irradiated cells, red color, compared with untreated cells, green color. Most genes have a little change, that is a yellow color. If you see that the red color DNA, CDKNIA, which is red, the gene expression is increased, whereas CMYC, gene MYC, which is decreased in the expression. So how it can be seen that? If you see that, if the green color gene is present in the normal means, it has to be present in the diseased condition also, na? So how they are interpreting? See, whatever you see the green color, those gene expression was decreased when you expose the cells to radiation, whereas when the cells are exposed to radiation, the gene name CDKNIA is overexpressed, whereas CMYC is decreased. So tomorrow, if anybody goes to a doctor saying that I went to space, am I affected? So tomorrow what they'll do? They'll isolate the cell down, just collect the RNA and then check the level of CDKNIA or else CMYC. Yes or no? Understood? Yes or no? Can I have response from everyone? Very, very important. Yes, Nitish, why not? Namita, red color and green color intensity is totally different, yeah. CDKNIA is the name of the gene. If you want to read more, read about that whenever you find time. It is some gene. How this is related to astronauts? In nowhere it is related to astronaut Mala. Just I want to give a situation. C5 doesn't find any gene, disease gene, Rajesh. If you see here, we are labeling the mRNA collected from the disease only with the red color dye and then green color dye. Okay. Yes, Mandira, you are correct. Any other questions? If the red color intensity is very less, which means that very little is expressed. It is not overexpressed. Yeah, it is very expensive. One test costs around like 2 lakhs or 3 lakhs based on what group of genes you want to express. How much time is required for probing? It depends upon the type of gene, type of set of genes. Maybe half an hour to 2 hours. Nothing is custom made. Everything you guys have to standardize. I didn't get your question, Gayatri. What is one-time processing is what? I didn't get your question, Nitish. You are asking about cost. One second. Show you how much it costs, but it is not loading. Okay, so I'm not able to find out where it is. Some problem. It depends upon the slot, depends upon the microchip, what you want to do. Is there any difference in analyzing different genes? For analyzing any gene in the same sample we use? It is not like what you think. It is so simple, Gayatri. See, for example, if we take the asteroid story, I don't want to analyze the genes which are not related to radiation, okay, because it's going to be very costly. Okay, so the thing is, so what I will do now, so during radiation, what and all can happen? For example, definitely the cell might be undergoing stress, isn't it? So what I will do now, I will select a gene which are related to stress related genes alone, number one. So once the cells undergo stress, definitely the cell death will be there. So definitely apoptotic genes. So I can select. So like that, based on the literature, I can collect what are the genes which are related to stress. I can collect 1000 genes, and then apoptotic genes, 1000 genes. Known genes. Those genes will be immobilized on the gene, on the glass plate or chip, like this. So I know that this part is apoptotic gene A, this is apoptotic gene B. I can read it. So I can choose. I don't want to go for a whole genome. You are isolating. So when the body is exposed to the radiation, I want to see what are the genes are responding, overexpressed, downregulated when exposed to radiation. That's it. Did you understand what I'm saying, guys? There are so many probes. Apoptotic probes are there, cell cycle probes are there, and then DNA replication cycle probes are there. I want to see, for example, if this person is treated with particular drug, what will happen to the DNA replication? So what I can do, I can collect all the genes that are related to DNA replication. After treatment, before treatment, I can check what is the level of the gene. It's very specific. It's very costly because each and every gene has to be synthesized, and then it has to be labeled. It has to be immobilized on the glass slide. Understood? You cannot, it's use and throw. Litika, I told that even anything can be done. Why not mutation? Anything can be done, provided you should have an idea what you want to do. Anything, but the cost matters. The cost is not a problem. Then you can think anything else. Anything. Any questions, guys? How is the sample and the control loaded on the chip? What is the answer for this, guys? What is the answer for this question? Exactly, Aarti. How the sample and the control loaded on the chip? This is the question asked by Sainika. No, yeah. Actually, this probe. This is your control, this is your sample. Both of them are just flooded. This small plate will be in the small tray. Both the cDNA will be flooded on the surface and then it will be incubated. Okay?